serious limitations like low nerve agent hydrolyzing activity and poor pharmacokinetic activity. We thought for recombinant human PON1 is like our human protein first synthesized in the liver, and when it associated with high density lipoprotein, then it will circulate in our body. So recombinant human PON1 can bind more than one molecule of organophosphate nerve agent like bioscavenger, also known as catalytic bioscavenger. So, but the development of catalytic bioscavenger against organophosphate nerve agent poisoning is accepted and validated approach. Towards this, different agencies like U.S. Army and Israel Army are working for this from 2001 onwards. So the solution, okay, the solution and scientific validity. So recombinant human PON1 has two problems: poor pharmacokinetic activity, as reported in literature. It can stay only 3 hours in human body, so and less nerve agent hydrolyzing activity. Towards this, we use the fusion protein approach to increase the half-life of protein. We use recombinant human PON1 with fused with non-recombinant human PON1 protein with other body proteins, and we use the some mutagenesis approach to increase the organ phosphate nerve agent utilizing activity. Then we got this molecule and we patented, and we got the trademark also, albumin peroxidase. So then we tested this molecule in HD rat animal model. So we use as a control a vehicle plus upon exposure of organophosphate. Now currently we are not using any nerve agent in the lab. So these are the very restriction chemical. Only defense can use, like in our country DRDO can handle it, and other defense agencies. So in our lab only surrogate is there, parathion derivative is there. So we use that. So upon exposure of peroxone, that is death. Then previously idea is like that ki Lbupon can be developed as prophylactic. Prophylactic idea is that if we injected Lbupon like other prophylactic agent in the body, it will circulate in at least time of 1 day, 2 day, and it can move, and the small operations can be done by our forces. So, but Lbupon we tried at 2 hours before prophylaxis, and then addition of the toxin peroxone, then mice will be protected. So prophylactic protection by Lbupon is there. We publish the paper of this. So limitation of Protexia are overcome by Lbupon. So work done so far for development of Lbupon. Okay, so we engineered the novel recombinant human PON1 variants. So panels of novel variants of recombinant human PON1, more than 30 variants are designed. Panel of variants are produced and characterized. One lead candidate are identified like Lbupon. So next, like clone and process development, we are producing this recombinant protein from E. coli. So clone develop and industrial feasible lab scale production process of Lbupon is also developed in our lab. So next step for we are went for characterization of this molecule. Lbupon exhibited enhanced OP hydrolyzing activity in our lab. So in vitro hydrolyzing activity, peroxidase, then Lbupon exhibited enhanced pharmacokinetic properties in SD rats. We reported 25.1 hours pharmacokinetic property in SD rats. Then we checked the Lbupon prevent the development of organophosphate poisoning symptoms and mortality in animal with control with recombinant human BUCHA on other approved drug for prophylactic and post-exposure treatment for organophosphate nerve agent poisoning. So then we don't have this nerve agent in our lab, so we send this sample to France Army, IRBF France. So they check the in vitro nerve agent neutralizing activity in buffer. So we got the results. So all G-type nerve agent hydrolyzing activity of Lbupon. We haven't reported this result anywhere. So POC established. So these are the patent and papers are reported from our lab, and these are the prototypes. So we are sending with our collaborators every time with these vials and all. So current TRL status is like 4, and cost analysis ratio, if we consider it like in our lab scale process protocol, so it is around 9,000 to 10,500 rupees we need for production of 1 milligram of protein. So what is road ahead? So currently we just done with the surrogate study. So we want to test the efficiency determination of Lbupon to prevent nerve agent toxicity in vivo. With currently we are working with DRDO Gwalior for this. So next step is with the dose optimization study with available chemical antidotes. With maybe some synergistic effect will be there. So we want to check that also. So next we obtain the dose, then we want to go for the non-human primates like monkeys, and then after that we will go for safety pharmacology, and simultaneously we will go for a production process and GMP level production of Lbupon, then final data generation and IND filing. Our expectation is like industries from joint research and technology transfer. So market potential, since there is this antidote against organophosphate nerve agent, there is not that much use market. But since the this is like it can come under emergency situation like COVID-19, like eye opener, we should prepare for next any uh, attacks and other things from for our nation's defense point of view. So thank you.